Chuan, Hsiu Chuan, this is actually your data, not the current data you talked about, but from your 2021 paper
Oh,
on
wow.
NGN2, when you really, I think that was one of the first reports. I think two came out together or near each other, showing that NGN2 wasn't this perfect cortical neuron thing, and PRPH is one of the markers you were using at the time for the sensory or even peripheral, depending on which level of PRPH positive cell you got. So this is a map of your time course in that data. And so this is the NeMO Analytics website that I've shared a couple times in this seminar series. And it's free and public. I can give you everything I show you today. I can give you links that you could explore on your own. So in this view, you can just type in a gene and see it. So for instance, this is PRPH. Here's EMX2. There's much less EMX2, but where you see EMX2 most, right here, if everyone can see my cursor, if we flip back to the PRPH, among the high expressing PRPH or later neurons, it's actually in the low area, of course, because these fates are kind of reciprocal, right? If you're getting one, you're not getting the other. So this is just looking at a single data set and a single gene. So if people have questions, please ask. If you want to look at particular things, please ask. But I'm just going to walk through and give you a tour of what we can do if we begin to get all of our data in a common space. So for instance, if we had the morphogen gradients and then the NGN2 and morphogen dosing experiments in the same place with that in vivo data, for instance, the Braun atlas we have in this environment too, what other things we could do. So maybe let me start here. So this is another single link, but instead of having just one data set, there are many. These are all in vitro neural differentiation data, mostly telencephalic organoid data. Again, you can type in a single gene. PRPH almost comes up blank, but interestingly, there are little pockets of it popping up. So it might be interesting to see what those are. If we look at EMX2, again, trying to look at the flip side, these organoids light up because, as I mentioned, these are mostly dorsal patterned organoids. And the kind of seed I want to plant for this group is, again, if we get all of our data in the same place and then begin to use things like perhaps gene lists that are associated with the morphogen gradients in the first talk, or gene lists that are associated with the individual dosing in the NGN2 experiment, the NeMO Analytics framework not only allows us to look at individual genes, but allows us to look at gene groups. So this, I flipped to another page now, is the same collection of data sets. These are many different in vitro data sets, the Uzquiano ones, the Canton ones from Barbara Trutline's group, Baduri from Kriegstein's group at the time. She has her own group now. But as you can see here, there are many, many, many that we have in here. These are more from Arlotta's lab. We have many from the Vaccarino lab. Alex Jordan, I don't know if you're still on, but your data, of course, are in here. And now, instead of having a single gene here, we can have any list of genes we would like. And that could be as simple as your 5 favorite genes. And in this case, we have S phase genes and G2M phase genes. So for instance, here you could see the S phase is right there. So of course, these are progenitors moving forward to those neurons. But you could imagine a whole list of other things here that might be chiron, XAV or chiron dosage and the genes associated with them. So they can be a simple list of genes with no weights, just 5 favorite genes or 10 favorite genes associated with a condition, or they can be more complex things like a genome-wide principal component that has a weight for every gene in the genome. And as you can see over here, we have a number of different mathematical implementations to project those gene lists into data sets. So as you can see here, S phase is lighting up a particular bunch of the progenitors. We'll see how fast our load time is now. But if I hit the G2M phase button over here, every data set on this page will now reload and show you where G2M phase is happening. So if you remember where the first one was, it looks like both G2M and S are up in here. So those are our cycling progenitors. It does look like there's a second entire population of cycling progenitors, and the annotations from the original authors show us what those are. So these are the IPs, the intermediate progenitors that are cycling. And back here in blue, these are going to be our radioglial cells that are cycling. And again, mostly what I want to do here is plant the seed for imagining if we bring together just these two things, they're kind of big things. One is all the data you're interested in. And then secondly is the gene sets you're interested in. And all of a sudden we can ask a huge number of questions from a huge number of systems. So I'm very happy to share these links when I post the next things on Slack and the emails to the group. I'll include them to show you how they work. And I really invite you to either use NeMo itself or ask me how to get your data in here or how to use it to explore what you're interested in, because we have a huge collection of data now for both in vitro and in vivo data on brain development. And for cortical neuronal fates, dorsal cortical neuronal fates, we have a nearly comprehensive collection. So pretty much any data set you can think of from the literature in those studies, that we have it. So we can really begin to, if you bring in a list of genes from your morphogen study, all of a sudden you can look at everything. Speaking to your question when Laura was on, I don't know if she's still here, I think she had to run. But what does this mean for in vivo work? For sure, it's great to have an in-depth bioinformatic analysis to compare one in vitro data set to one in vivo data set. But with these tools, we can really look at many, many more. What it looks like in the first trimester, what it looks like in the second trimester, what directed telencephalic organoids look like versus NGN2, and so on. So I invite you to interact with me or with the NeMo website to get more data in here and to get these tools into your hands to explore this more. And we can do a little bit more of this next week after the session, because again, we'll be in morphogen land. But with that, I want to thank you guys again for coming. I'm very happy to explore more or take questions here, but I know we are at about just 2:15.
Carlo, can I just ask you one
Sure.
quick question?
Yeah.
So with NeMo, can you just set up your own, list of datasets that you want to compare?
Yeah, so for instance, here I'm logged in. To look at all this stuff, you don't even need an account. But if you want to upload a data set or upload a gene list, you do. So here, under my name here, I know this is small, so I'll read them. There's Dataset Explorer. So that could look at any data set I have uploaded or any data set anyone anywhere has ever uploaded, as long as they make it public. So that's there. And the second thing is Gene Cart Manager, which is the same, but for gene lists. So if I click on that, this will populate in just 1 second. This is, again, just like the data, it'll bring up any data set or any gene list I have ever uploaded, in addition to any data set anybody or any gene set anybody else has uploaded to NeMo. So I have myself, I've brought in 137 different gene lists. And in fact, I think I have data up for one of them here. We can flip to this one. So this is, we can go to Hsiu Chuan's data again here. So this is her, the NGN2 data from some time ago, but now it isn't colored by a gene, it's colored by retinoic acid induced genes. So a gene list from a totally different study. And here you can see at this day 5 time point, there is an entire packet of cells that have particularly high retinoic acid. And if you remember, let's see if we can. This was the EMX high, and all of these were the PRPH high. So then there's even a subset of PRPH neurons for some reason. I don't know if it's difference in fate or difference in differentiation level going further down the road, but there's a set of those PRPH neurons that are higher in retinoic acid response gene expression. So it's trying to put together two very, very simple things to be able to ask lots and lots of questions. Get the data together, and then get your sort of hypothesis-driven or question-driven gene list together. And it really does depend on what data is there, obviously. And that's why I'm really throwing out the invitation and hope people start to get their own data in here, because that's when it becomes powerful, when we can really look across a lot of data.
Great, thank you. So, and one last thing, can you just download it from NeMo? Is it like?
Absolutely. So
that's
yeah,
amazing.
it's
That's
insane.
amazing.
So every single data set you see here, if I click on this button, download a H5AD version or
Okay.
download 3 text files. So any data set you see, bing, 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 download, 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 done.
Wow, wow, like congratulations, this is amazing.
I'm really hoping that a lot of people start to use it because, you know, we have kind of a gap between the paper where all the details are,
Yeah.
but of course you can't interact with it, and then the repository where the data is there, but you got to go download it. You got to make sure the metadata are there. You got to curate the metadata, ask the authors, oh, you forgot to put the time or the samples in there. Okay, thanks. Now I download them. Oh, it's in a different, you know, format from my other one. So looking at them all, you can do right in NeMO, and anything you see in NeMO, you can just click and get.
Do you have an estimate on how many data sets you have in there in total, or number of cells?
So for the neocortical development area, we're writing this paper right now for the BICCN developmental brain package that's going to be submitted. We did a count. So we're nearing 200 studies, so 200 papers. That's somewhere, if you actually look at how many matrices, samples by gene matrices that we have across those papers, it's at like 1,000 or 2,000. And between all the samples, we're at 20 or 30 million cells / samples.
Brain specific data, you see,
All
right?
neocortex, not even just all neocortex. So it's all, like I said, it's almost every neocortex study that you could think of. There's not too many out there that we don't have.
Okay. Thank you, Carlo.
Well, thanks, guys.
Thank you.
Thank you, Soraya.
We are interested in the Vaccarino's lab to upload our data set in the NeMO.
That's wonderful.
So,
I'd be very
Alex.
happy to work directly with you. We're trying to make that process easier and easier. If you work in R, we have R scripts that can take your matrices as arguments and package it up and add all the identifiers to it to make that easily possible. So absolutely, I'd love to work with you to help you do that and get more data in here.
Yeah, thank you.
You're welcome. Thank you. Thanks, everyone. See you in a month. Ciao.
Bye.